my dear students. Welcome to lesson 6, and we are still looking at the chemicals of life. And in this particular lesson, we are going to conclude with carbohydrates, where we are going to do the chemical tests for different groups of carbohydrates. And group number 1 is the reducing sugars, and we are going to do tests for reducing sugars. The reagent for testing reducing sugars is Benedict's solution. That word Benedict starts with a capital letter because it's the name of the person. It's a noun. So Benedict solution, and the color of that reagent is blue. It's blue in color. Or we can use Felling's solution or Felling's reagent, which is also blue in color. The condition for this reaction is boiling. We should boil so that the reaction can proceed and we see the changes. So when we are testing for reducing sugars, what is the reagent? The reagent is Benedict's solution or Felling's solution. And what is the condition? Condition is boiling. So let's look at the procedures, the observations, and the possible conclusions here. So possible procedure here is to 1 centimeter cubed of a food solution, add 1 centimeter cubed of Benedict solution and boil. So the instruction can appear like that. But at this level, you are expected to write your own instruction, your own procedure, because we should be able to do that now. Yeah, so you have to write it. You have to report in a reported speech. So how do we observe? The observation is a colorless or turbid solution turn to a blue solution, then to a green solution, to a yellow precipitate, to orange precipitate, and brown precipitate on boiling. So what are we talking about here? We say that to 1 centimeter cubed of a food solution, add 1 centimeter cubed of Benedict solution and boil. So this is what we observe. If the solution that you are testing is colorless, then you say a colorless solution. If it was turbid, you say a turbid solution. So you say a turbid solution turned to a blue solution. That is on addition of Benedict's solution. As soon as you add Benedict solution, that is solution, whatever it is, will turn to blue solution. And then on boiling, it will turn to green solution, yellow precipitate, orange precipitate, and finally to brown precipitate. However, it can stop at any color there. You don't have to force it to reach the last color. You have to accurately report what you have seen. So how do you conclude? You say either little, moderate, much, too much reducing sugars present, depending on the color. For example, we say that little reducing sugars are present when the blue solution turned to green solution. And that is the final observation after boiling. It doesn't change anymore. And then when do we say that it's moderate? We say that it's a moderate when it turned from blue solution on addition of Benedict solution, then on boiling it turned to green solution, and then to yellow precipitate. That one is moderate amounts of reducing sugars present. And then much reducing sugar is when it goes to orange, and too much when it goes to brown. Or orange and brown can be generalized as much reducing sugars. No problem for that. So those are the possible deductions or conclusion that you can make. What about if the test is negative? A colorless solution or a turbid solution turned to a blue solution, which persists on boiling. So if a solution turned to a blue solution on addition of Benedict solution, and then on boiling it remained blue, meaning reducing sugars are absent in that case. So that is the test for reducing sugars if you are using Benedict's. If Felling's solution is used, The change from blue to orange precipitate if reducing sugars are present. It remains blue solution if they are absent. So the test is the same. The observations are the same and the deductions are also the same. It's only the reagent name that changes. So what are those reducing sugars that we are talking about? In actual sense, the reducing sugars we are talking about is glucose, which is found in fruits, in grapes, for example. When a fruit can be is crushed, for example, pineapples can be, or grapes, you can squeeze juice from it, and that juice contains some glucose. Fructose is found in edible fruits as well. Galactose in milk usually. Maltose usually in germinating seeds. So any of those samples can be crushed, and you can be told to test for reducing sugars. You will find them present. What will vary is the amount. So, but that is the source of the reducing sugars that you are given in the laboratory. They are got from those samples there. So I want us to watch this video here showing us how some students did tests for reducing sugar. So you can watch it and appreciate how this test is done and how the colors change. Usually the colors disturb most of the students during these tests. So let's start afresh the video. Are we there?
Add about 2 cm³ of glucose solution. We can use a dropper to measure exactly 2 cm³ into the test tube first. This is followed by adding an equal volume of Benedict solution, and you notice that we will change another dropper so as not to contaminate the solution. Again, we use a dropper to measure exactly 2 cm³ of Benedict solution and add it to the tube. Shake the test tube to mix the contents. Then we will heat the test tube in a beaker of boiling water. First, we use a test tube holder to secure the test tube. Then we put the test tube into the beaker of boiling water, and we make sure that the water in the beaker is higher than the solution in the test tube. We will leave the test tube in the beaker of boiling water for 2 to 5 minutes. Observe the color change. The original color now is blue.
So in each case, we should be able to make deductions. You can see the color changes that are taking place. If the final color change is brick red, what does it mean?
Once it turns into a brick red precipitate, we will off the Bunsen burner.
So usually we must be very observant when we are conducting this test, because
And now you can see the orange red precipitate.
in most cases these colors change rapidly. They don't take so long. So if the student is not so much observant, you may end up of the colors, and yet the color changes took place. So you must be very keen. don't remove your eye off the beaker, and I mean the test tube, until you see all the colors, so that you can report accurately. So let's proceed. Test for non-reducing sugars. I should notify you that there is no direct chemical test for non-reducing sugars. There is no direct chemical test for non-reducing sugars. But what we can do is to first convert them to reducing sugars, and then from there, then we can now test for the reducing sugars. We first break down the non-reducing sugars to reducing sugars, and then we test now for reducing what? Sugars. So that is what we can do. Otherwise, there is no direct test that we can perform to test for reducing sugars directly. So we can see from here, our procedure is a bit longer because it involves the process of hydrolysis, as we shall see. So let's go through the procedure together. To 1 cubic centimeter cubed of a food solution, add 1 centimeter cubed of dilute hydrochloric acid and boil. So that's the first step. You get your food solution, add dilute hydrochloric acid, usually in equal amounts or slightly less, and then you boil. Then after boiling, you cool under water, and then add 1 centimeter cubed of sodium hydroxide solution, followed by 1 centimeter cubed of Benedict solution, and boil again. So in our observation, it is similar to that of the reducing sugars. Your colorless or turbid solution will turn to blue solution, and then to green solution, to yellow precipitate, and then to brown precipitate on boiling, signifying either little, moderate, much, or too much reducing sugars present. It is actually reducing sugars present, not non-reducing sugars. Our conclusion, perfect conclusion, is either little reducing sugars present from hydrolysis of non-reducing sugars, or moderate reducing sugars present from hydrolysis of non-reducing sugars, or you can say the non-reducing sugars hydrolyzed to reducing sugars. So any of the two, but you must indicate that the non-reducing sugars were first hydrolyzed to form reducing sugars. You discard that observation of that conclusion of non-reducing sugars present. It is not accurate. And then colorless or turbid solution turned to blue solution, which persists on boiling, meaning the reducing sugars are absent. Or you can perfectly say that non-reducing sugars not hydrolyzed to reducing sugars. Non-reducing sugars not hydrolyzed to reducing what? Sugars. Not the way it is there. So that is how you would test for non-reducing sugars. We are going to find out how these tests are done. We can also look at this YouTube video, and then you watch and see how the sugars are first of all hydrolyzed to reducing sugars, and then finally the reducing sugars are tested using either Benedict solution, one of the two.
Okay. Test for non-reducing sugar. You need test tube rack, 3 test tubes, you need some cylinders, you need some, again, Benedict solution, hydrochloric acid, sodium hydroxide, and some sucrose solution. Okay, let's add 10 ml for our test control, the water. Then we have our sucrose solution, 10 ml. And we're going to add a few drops of hydrochloric acid in the test control. This one we don't put any hydrochloric acid, we leave it aside. We're going to take those 2 test tubes and we put it in the water bath for a few minutes. You put your 2 test tubes, test control, and the sucrose or sugar solution in the water bath for a few minutes. While they're cooling down, you have to neutralize the hydrochloric acid, so you need to add a few drops of sodium hydroxide. Drops. Then again, you don't touch that one here. Now it's time to add the Benedict solution. Add a few drops in each test tube. And we take those test tubes and put them in the water bath for a few minutes.
So we know why we are putting it in a water bath.
So we did.
It is to provide the condition of
test with non-reducing sugar. As you can see, this is a control with water, so no reaction. Here, this is the test tube with sugar and hydrochloric acid. So at the end, you get a reaction because you broke down the disaccharide into glucose and fructose. And that one is basically sugar, so sucrose, with no hydrochloric acid.
Okay, thank you very much. That was great, and we have seen how the tests can be done there in our laboratories. So we want to explain some of the processes there. When boiled with dilute hydrochloric acid, the non-reducing sugars break down into reducing sugars. So the reason why we added dilute hydrochloric acid is to break down the non-reducing sugars into reducing sugars. Like we said earlier, that non-reducing sugars have to first be broken down because they do not have a direct test for them. And then why do we add sodium hydroxide solution or a sodium bicarbonate, sodium hydrogen carbonate powder? It is to neutralize the hydrochloric acid because Benedict solution works best in a neutral medium. So examples of the non-reducing sugars we are talking about is sucrose, which is found usually in sugarcane, in cane sugar, the table sugar at home, and then lactose is found in milk, and also other non-reducing sugars. Those are the major ones. Let's proceed to another test for starch, popularly known as the iodine test. This one you must have familiarized with it at O level. And we have the procedure, observation, and the conclusion. To 1 cubic centimeter cubed of food solution, add 3 drops of iodine solution. What would you say if you are the one writing the observation? I mean, writing the procedure yourself and you are writing a reported speech. What would you say? Probably you could say to 1 cubic centimeter of food solution, X or Y, whatever it was added 3 drops of iodine solution. Maybe you could say that. That would be much more better. It looks like it's a reported speech. But however, this one is the one, it means this one, somebody is instructing you to do it. To 1 centimeter cubed of food solution, add 3 drops of iodine solution. The observation would be, in case the starch is present, a colorless or turbid solution turn to black or blue-black or blue solution, or brown solution with black specks. Remember, only one of those observations can be realized. So you write only one that you have seen, and only one observation of the original solution can be seen, either colorless or turbid or milky. What you have seen is what you write. You don't say turbid or, it means you did not see. So you can say a colorless, or you can say a turbid solution turned to a black solution. Or you can, if that is what you saw, Or you can say a turbid solution turned to a blue-black solution. Or you can say turbid solution turned to a brown solution with black specks. Then in your conclusion, if the solution turned to a black solution, then you can say much starch present. If it turned to a blue-black, then you can say moderate starch present. If it was a brown solution with black specks, then very little starch present. So depending on what you saw, you need to report accurately. What about if the colorless or turbid solution turned to a yellow or brown solution? What would it mean? It would mean that starch is absent. Our dear students, remember you are the one writing all this procedure, you are writing observation, and you are writing the deduction. So you write exactly what you have seen. Here you have seen a variety of possibilities simply because it is theoretical. I'm giving you the theory of this information. But when you are writing it, you write exactly what you have seen. And I know this one is much more simple for you. So we can watch this other video to see how we can test for starch, just in case you forgot your O-level information. Remember, in test for starch, the reagent is iodine solution. Yeah, that is the reagent that we use for testing for starch, and it's brown in color. Thanks to University of Arizona.
In this video, I will demonstrate how to use an iodine test solution to detect the presence of starch. Starch is a type of carbohydrate that is found in large quantities in foods that contain wheat, corn, potatoes, or rice. Many of the foods you will be analyzing in lab contain large quantities of starch, and an iodine solution can help identify whether or not you successfully removed all the starch from your isolated sample. For this demonstration, I have two vials. The one on the left is pure water, and the other is a solution. A single drop of the iodine test solution each. The vial on the left turns a yellow-orange color, and the one containing starch turns a black-blue color. This iodine test works well in aqueous solutions, but if the solution you are using is a viscous organic liquid such as oil, the test may not work as well. To test a lipid solution, you should dilute it with an organic solvent first and then add the iodine test. While the iodide will not dissolve in an organic solution, it is for the presence of starch. This organic solution does contain some starch, as indicated by the small drops of black liquid at the bottom. Hopefully you found this demonstration on how to use an iodine test solution to be helpful. Good luck and have fun in lab.
Thank you very much to the University of Arizona for that wonderful video. So you can see now how we can test for starch in our laboratories. Remember, we are using iodine solution, and iodine is brown, but when you drop it to a solution containing starch, it turns to either black or blue-black or blue or brown with black specks, depending on the quantity of starch. At our level, we are also encouraged to quantify the foods that we have found. That's why we say moderate, little, much, and so on. It's very important because in future you would be a medical doctor or a laboratory technician in the hospital, and you will have to quantify the prevalence of the disease or the quantity of the disease parasites that are available so that action can be taken. That's why we do that at this level. So we can also watch this other video to see the different food samples and comparing the amount of starch present in each. Don't forget that this is a very simple
Testing
experiment.
for starches in food. We need a balanced diet to give us the energy we need to work and play. Starches serve as a quick source of energy. But how can you tell if food is a starch? We're going to answer this question in just a few minutes by performing a simple experiment. Before we get started, remember, never touch or taste anything in the lab and always wear your lab coat and safety goggles. Okay, let's get started. Alright, for this experiment, we're going to need a small bottle of iodine and a paper towel. We're also going to need 10 small samples of different food. I'm going to place all the samples of the food on the paper towel. Then I'm going to take a drop of iodine and place it on each sample of the food and see if it's colors.
Iodine is a reddish-brown liquid that turns blue or black on a starch. Be careful with the iodine. It can stain your clothing. After all my observations, I'll record the findings in my data table. All right, here we go. Okay, all done. I'll clean up and wash my hands. So just as I suspected, the potato, the pasta, and the corn all turn black because they're starches. But I also had some surprising results. Looks like I need to make my meals a little more balanced.
Wow, that's great. So you can see that even the food that we eat contains starch, but in varying amounts. So you may find that some foods have more starch than the others, depending on the, as you saw the colors there, the deeper the color, the more the starch available in that food. You can do this when you come back to school with iodine, so that we can do a similar experiment. So we have tested for reducing sugars. Do you remember the reagent that we have used and the processions and possible conclusions? We have also tested for non-reducing sugars, and then we have tested for starch. In other words, we have tested for most of the common carbohydrates that we have. I want you to go and write in your book a summary of how we can test for starch. You write the procedure, the possible observations, and the possible deductions. How do we test for reducing sugars? How do we test for non-reducing sugars? Write that in your book. It is one of the ways of mastering these tests, so that you can be very perfect when you come back to school. Thank you very much for being good students. Meet you in the next lesson.